more clean and better fed. Thank you, Warner. I'd also like to thank amfAR for the events of today and for enabling all the science. It's surely going to happen over the next 4 or 5 or more years. I also like to thank all of you for attending today, especially the members of the community, and most especially those of you who've been involved in our research in the past and or who wish to continue being involved in the future. There's lots to be done, and we have never done anything in this city without you. I've been here for 33 years. I've watched the epidemic during that time. I've been involved in it. Like you, I've seen the pain. I'm thrilled now to even think about the possibility of putting this to rest. And I think, you know, with the kind of cooperation, collaboration we have with the community and with the scientists here and elsewhere, that this is something that we can do, and I hope we can do it sooner than later. The small part that I'll be involved in, I'll talk to you about today, is called Module C for charting the persistent reservoir in patients that are treated. Let's see if I can work this thing as well as, yay. So put another way, in those patients who are treated with effective antiretroviral therapy, who have no virus circulating in their peripheral blood, where does persistent HIV live? And that's really what we want to bore down to in this next set of points about the problem that you heard about in the past, which is that even though these potent medications can cause the virus to disappear from the peripheral blood, essentially, and it does, of course, lead to the health of patients in a way that we never would have imagined in the 1980s. Loss of virus in the peripheral blood is not the same as eradication. The virus is still there. And where does it live? It lives in those roots that Rowena was talking about. And those roots basically are the roots of your immune system, which are made up of organs that are spread throughout the body and which harbor the virus. And until we get rid of the virus in those organs and in those roots, it's unlikely that we'll be able to effect a functional cure or, you know, if we could, a sterilizing cure. To tell you a little bit more about how we're going to do this, I want to give you a quick trip through the immune system. So these roots, these actually are organs and parts of the immune system that are found throughout the body. They are there to defend your body against incoming infectious agents like HIV. Some of them are organized into compact structures that you can actually hold in your hand. The lymph node, the spleen are like that. Others are spread diffusely across the skin, the lung, the gut. You know, they're as part of the innate immune system that you heard about to respond very quickly, to agents that come into the body. And the thing that's common to all of them is that they all interact and they all have CD4 positive cells within them of different types that can be infected by HIV, and therein lies the rub. HIV basically goes in very quickly, it infects and kills cells that are there to defend you against HIV. And this is the problem we've been dealing with for the past 3 decades, and the problem, in fact, that we will be dealing with when we try to eradicate HIV from patients who are treated. We've known for a long time that lymph nodes can harbor HIV. I bet, you know, Stephen probably showed lymph nodes underneath his arm like this when he was first infected. This is the kind of thing that happens during acute infection. And that node that one can feel is actually a lymph node that's been infected by HIV and that is initiating an innate response which causes inflammation and pain. And that's something which is a sign of acute HIV infection. We've looked at these nodes carefully, mostly by cutting down a surgical procedure that's minor but still more painful than drawing blood, to find that they're small, usually, about the size of a pea. And if you open them up, you find that they're very elegantly arranged, somewhat like a home. There are different zones that have different parts within them. There are some parts that have T cells, like CD4 positive T cells that are found, and other parts in which B cells reside. These are cells which you simply need to know are the ones that are specialized to make antibodies. They live in different parts, they communicate with one another, and they do so in order to have functions that are distinct. So virus will actually move in through specialized parts of the lymph node and thereafter interact with T cells to create a T cell response that might fight against the virus, usually not quickly enough. Those T cells can actually help B cells that are in the B cell follicles to make antibodies that might neutralize the virus. Again, usually not quickly enough. And that's happening usually for other infectious agents in a way that does work, but in the case of HIV, that's not the case. These structures work well, though, and they're found throughout the body in more or less the same kind of structure, the same kind of house-like structure in the gut, which is probably the home for about 90% of the CD4 positive T cells that reside in the body, and which is a major focus of HIV infection, and therefore likely a major source of persistent virus. And then importantly, they're also found in the female reproductive tract. This is probably the main source of HIV entry into women that are infected, and there are many of them infected around the world, and we think likely it is the source of persistent virus that exists after antiretroviral therapy is started. So given this complex immune system, what we're trying to do in this module is to understand which organs are infected, which cells within those organs are infected, and is what we find in some patients translatable to all patients? Or in fact, are there differences between men and women, those who are older and younger, and those who might have been started on therapy at different times after infection? We have a wonderful team of people who's working on this. They bring many different strengths to the fore. I'll be talking a bit about some of the work that they're doing. Zoltan Lazic, working with Raja Hussain and Josh Vesquez, will be working in the beginning to optimize and standardize techniques that will simply allow us to see virus, that 1 in a million that Warner mentioned, and ask the question, where does it reside and how does it look? And they'll be doing this in multiple tissues, in patients that do have suppressive antiretroviral therapy on board, and using a number of techniques. I'll show one of those techniques now so you can just visualize exactly how we're going to be doing this. It's not as hard as it may look, but in fact getting the tissue and getting this technique to work has been something that's been quite formidable. If you imagine, this is a technique that we call DNA scope and RNA scope, depending on we're looking at the DNA of HIV or the RNA of HIV. And what we can do if DNA is integrated, as shown in red here, into the host genome in that middle circle, which would be called the nucleus of the cell, is to add 2 probes that very quickly come down and specifically bind to the HIV that's there. And then if they're linked, they actually can be seen by the light microscope or by a fluorescent microscope in a way that they could not be seen before. So too, if there's HIV RNA in the cytoplasm, there are probes that we can bind specifically to it, and once again in ways that can be seen by the light microscope or the fluorescent microscope, it would tag those molecules of HIV. What that allows us to do is in a sea of cells, this is in a lymph node, all of these cells that are blue are uninfected. These cells here that are red are showing evidence of HIV RNA, is shown here in more high power. And that is something that would be very difficult to do by other means in a broad survey of organs. In more recent work, it's possible now to identify what those cells are. So if you can see HIV DNA recognized by these purple dots inside the cell, this green color around it shows that the cell is a CD4 positive T cell. Likewise, using a similar kind of trick, it's possible to see that in this particular cell where there's a piece of HIV DNA within, that there is evidence that the cell is a macrophage. And this kind of tagging of the cells can be done for the multiple cell types that are present so that we can identify not only where the HIV resides, not only whether it's there with HIV DNA and RNA, but in which particular cell type it's found. Our goal over the next years is to standardize these methods and to be able to apply them to large numbers of individuals so that we can see in Rowena's picture here, where the grass is cut, exactly where the weeds reside. To do that, Josh and Zoltan and Raja will be working with Peter Hunt and Shomi Sanjabi, Nadia Ron, and Steve Yuckel to define persistent HIV in different organs from different people. Now, most of the people that have been studied to date in the epidemic, it's 30 years in, come from the United States and Europe. Most of them are Caucasian. Most of them are men. Most of them started out younger but are now middle-aged and older. And we essentially know nothing about what's going on with HIV in other groups around the world. So our intent here is to create a large group of individuals that are studied. We will complement this with other groups that are found in other studies that we're doing. We'll be looking specifically at men and women, knowing that in the way the epidemic is progressing now, of the 37 million individuals that are infected, about half of those are women. We'll be looking at men and women that are younger and older, knowing that in fact most of the new infections are found in younger people, not older people. and younger people behave differently than older people when it comes both to the way that the immune system works and probably to the way in which the virus can replicate. And finally, we want to look at those that are treated early with antiretroviral therapy and those that are treated late. Early is the way that we want this cure to work. We want to find people immediately after they're infected, treat them early with antiretroviral therapy, and then come in with a cure. But we haven't until recently been able to study individuals like this because it hasn't been until the past 4 or 5 years that such early treatment was really offered to many people. We'll be doing this in a number of different ways, looking at a number of different organs. There'll be biopsies taken of both lymph node and of gut. This is a picture that's schematically meant to show a lymph node. But basically, these organs can be removed by biopsy, and cells can either be isolated from them for interrogation by a variety of techniques that use single cells for the analysis of HIV and function therein, or underneath the microscope showing that technique that I showed you earlier called DNA scope and RNA scope. We're interested also in looking at cells that move out of the lymph node into tissue spaces found in the lung and in the skin and the gut and elsewhere. These are called tissue resonant cells. They're not something that you could biopsy with or see in a lymph node. Instead, one has to look at the diffuse tissue spaces that are found in these organs. And not least, we'll be looking at the female reproductive tract using the elegant virologic assay that Warner Green mentioned earlier that Eric Burden has developed. Nadia Rowan will be asking the question, where in the female reproductive tract are there cells that can be latently infected? And what do they look like? And how can we attack them? At the end of the day, our goal is to figure out where this virus lives. We want to know what the anatomical locations are in different parts of the body. We would hope to complement this with the full body imaging procedures that you heard about earlier and that you will hear about next. We want to know which cells the virus resides within these organs. And finally, we want to know which people, in which people the reservoir can exist in different ways or the same way. And if there are such differences, to pinpoint them so that they can be defined in a way that will help us to better tailor treatment and cure. If you go back to that nice diagram that Rowena showed earlier, if there are 3 main strategies, replacing by cell transplant or controlling with a vaccine or shock and kill, we won't really know that these methods are working unless we know where the virus lives and we know that the interventions have actually caused the virus that lives there to go away or to be substantially reduced. I'm excited about the possibility to be doing this work. I think, it has been said a number of times, it's not going to be a slam dunk. It's going to take years in the making, but I'm confident given the collaboration that we have here now, the resources that we have here now, and all of you working with us, that it's going to be something that we can do.